Last time on Office Hours, we looked at how to label ants with barcodes. Today, we're going to be looking at Andreas' experiment. Andreas is a postdoc here in the Hughes lab. And he's studying how density affects social food exchange in ants. This is quite a complex topic, so we've brought in Matt here to help translate this from science to English.
Hey guys, I'm here to de-jargonize all the cool stuff we're about to see. It's pretty exciting, so let's get to it.
Let's do it. So Andreas, we're interested in learning more about your experiment. What can you tell us about your goals for what you're working on right now?
So in general, what we want to understand is how societies work, and what we're using to understand better how complex dynamics function within a colony. For example, we use ants as our model system. So what we are doing is changing the colony itself. We're changing the amount of space they have in which they live, the nest size, as you will. So this will allow us to not only know if they're more active or less active, but also how food, which we're mostly interested in, will spread throughout the colony. And this will allow us to make models and spatial statistics to analyze potential for disease spread as well. Not only how food will spread through the colony, but also how diseases will spread. Because like food, things like viruses or fungi would also spread via direct interactions among individuals.
So the general purpose of this study was checking how density influences the spread of food throughout the colony. How fast can colony members that come in from the outside, basically foragers, they collected food to come back to the colony, how fast can they transfer the food to the nest workers and eventually to the queen. The queen is basically the big ant you can see right here, and she is acting like the reproductive organ of the whole colony. So she's producing all the future workers and all the offspring. And the workers have different roles in the nest. As I said, the foragers, they go outside and bring food back. Then you have nest workers or nurses who take care of the brood. And you might have guards at the entrance around this area who then pay attention that nothing bad happens, no other ant species or parasite comes in. In general, we look at every interaction that the ant colony has within the colony. We are mostly interested in one of the most intimate interactions, which is trophallaxis among workers. And trophallaxis is basically the transfer of food. So we basically have here two ants interacting, and you can see they're transferring food from one individual to the other one. And you probably don't know why this is important. If you don't know how the big colony, a big society functions, you have certain workers that go outside, that are foragers, will collect food, and then bring food back inside to the workers that stay in there, with the nurses, or the guards, and eventually to the queen, who is basically acting as the reproductive organ of the whole colony. And in general, we can then, because we have all workers labeled with barcodes, as you can see here, know exactly who was outside, who was foraging, with whom they interacted when they came back in, and eventually how long it took to spread food throughout the colony.
Where else do any other universities in the world do research like this in depth?
Yes, there are other universities, of course, that work on similar topics and that use automated tracking to analyze how food or diseases are spread throughout the colony. The great thing about our study is that we are actually doing it with the human eye. So error rates that result from tracking, like sometimes computers say, okay, they're like individuals are close together in that angle, so they're probably trophallaxed. We actually know that they trophallaxed. So that's the great thing about our data set, is that the quality is really high because we have the human eye actually tracking it.
Cool. And that's what Kevin is doing
This
here?
is what Kevin is doing right now.
So Kevin at the moment is labeling who and which individuals are where in the colony and writing down all the codes. He knows which individuals he needs to track and follow next. So his next step, what he's going to do is pick out an individual, like, let's say for example this, and then follow this individual throughout the whole video and write down every single interaction it had with other individuals. So he's going to write down the start and end point of every transfer of food that this individual had. So eventually he will know who interacted with whom during the whole video. And the great thing about this is that so far no other people have ever done this kind of quality for that amount of time. We have several hours that we observed. So we're going to be able to build a social network that shows us who is connected with whom, to what degree, for how long they interacted with each other. The good thing is that we are not using machines that a lot of other labs did, but we are actually using the human eye, which is far superior because we can tell exactly if they were trophallaxing and not simply say, yeah, they were close together in a certain angle, so they're probably trophallaxed. We actually know that they trophallaxed because we can see it, and we have a lot of undergraduate students that perform exactly this work. So the quality of data that we get is immense, and no other one person or scientist has this kind of quality in data.
Wow.
Yeah, what Kevin is currently looking at is a video that is recorded at high density. So we had two different treatments that we were analyzing our colony with. We wanted to see how different space, the amount of space that workers have influenced their activity and the trophallaxis of food. So as you can see here, there's a lot of ants in the colony. Density is quite high. So we want to know if that influences how fast food will spread and how many interactions they have. In another videos, we have basically 4 times the size of this space for the same amount of workers. So we'll know how density affects the interaction rate and also the social network that we can later build based on the results that we get.
What have you found out so far? Does density affect?
Oh, you will have to wait and see. So far, I don't want to give anything away.
Suspense.
Oh.
Yeah.
Say hello.
Do you want to have more lines from here?
We thought that we were going to just get you like far away and then have